Hello, everyone. Welcome to today's live broadcast, Simple, Fast, and Accurate Microbial Identification by Sequencing. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals, and sponsored by SciEx, placing the power of life-changing answers into the hands of those who care everywhere. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Dr. Jane Luo. Dr. Luo is a staff application scientist at SciEx, responsible for developing genomic applications on SciEx Capillary Electrophoresis product lines. Within her role as an application scientist, she works closely with key opinion and thought leaders to gain insight on the relevance and utility of various genetic applications. Prior to joining SciEx, she was a chemistry development scientist at Beckman Coulter, who led development of several reagent and chemistry kits used on the GenomeLab GeXP genetic analysis system. Previously, she was a scientist at Allergan working on anti-hypercholesterolemia drugs and an assistant adjunct professor at UC Irvine studying anti-VEGF therapy for human pancreatic cancer. Dr. Luo received her PhD in biochemistry from Hunter College of the City University of New York and received postdoctoral training at Cornell University Medical College and Harvard Medical School and Children's Hospital in Boston. I will now turn it over to Dr. Luo.
Thank you very much, Judy. Good morning, good afternoon, and good evening. Today, I would like to talk about simple, fast, and accurate microbial identification by sequencing. Why is microbial contaminant identification important? Well, if food is contaminated with bad bacteria, people will get sick when they eat it. If patients take the pharmaceutical drug products that is contaminated, they will suffer adverse effects or complications. Nowadays, we all try to live a healthier life by doing something good to ourselves, such as taking probio pills that will help us to improve our digestion. Each probiotic pill contains 10 billion bacteria. Can you imagine what will happen if that probiotic pill is contaminated with bad bacteria? You can get really sick. If products are contaminated with microbes, manufacturers will have compliance issue with FDA Guideline and ISO standards. Let's take a look at the impact of microbial contamination on quality. If the starter culture of a milk product, such as yogurt, is contaminated with a different microorganism, the yogurt may not taste the way you like. Same is true for beer production. If product is contaminated with microorganisms, they usually have to be scrapped, increasing the product cost. If contaminated products were not caught during QC testing and released to customers, they would have to be done with a recall. And any recall is bad for any company because it projects a very bad image for the quality of the products. I think we all understand and agree that microbial contaminant identification is very important. An FDA guideline for industry released in 2004 for industry on sterile drug products produced by aseptic processing, FDA recommended that any contaminated unit should be considered objectionable and investigated. The microorganisms should be identified to species level. So how do we identify microbial contaminants? First, let's look at the traditional methods for microbial identification. Most of them are based by phenotypic differences of different microorganisms. The culturing method is very common. The microbes are grown on different types of agar plates. This method is very inexpensive and easy to do. Gram staining can be used to determine if a bacterium is gram-positive or gram-negative. There are many different types of biochemical analysis available, from individual test tubes that contains different carbon source to plastic strips with individual test solutions packaged in a bubble wrap format. Some vendors offer 96-well plate with different carbon sources per well. Other vendors provide semi-automated or fully automated and miniaturized tests. These tests are usually very straightforward to follow and easy to perform. There are also many immunological tests available for some of the organisms. Let's look at the limitation of traditional methods for microbial identification. Very often they cannot identify microorganisms down to species level. The culture method usually takes a long incubation time. They're incapable of differentiating highly related species. Usually they are labor intensive and not automation friendly. The gram staining are prone to operator error, not automation friendly. The biochemical analysis usually cannot differentiate related species with similar biochemical properties, and very often they are inconclusive. The immunological tests are not available for all microorganisms. So you may want to ask, any other methods? How about the test based on molecular biology? Well, that's exactly what I'm going to talk about today, microbial identification by sequencing. Which gene to sequence? We recommend sequencing ribosomal RNA gene. Why ribosomal RNA gene? Ribosomal RNA is an important molecule which is part of a ribosome. Ribosomes are protein factories in all cells. Due to its important function, ribosomal RNA gene, also called rDNA, is the most conserved DNA in all cells. The rDNA sequence from distantly related organisms can be precisely aligned to readily identify the true differences between them. In prokaryotic microorganisms, such as E. coli and Streptococcus, there are 3 different types of ribosomal RNA. They are 16S, 23S, and 5S. The 16S rDNA, 23S rDNA, and 5S rDNA are all within the same operon. They are transcribed together. The region between the 16S and 23S is called IGS1, or intergenic spacer region 1. And then the region between 23S and 5S is called IGS2. Some other scientists also call IGS in a different name as ITS, internally transcribed spacer region. Both IGS and ITS are correct terms and used by different scientists in their publications. Among the 3 ribosomal RNA, the most conserved one is the 16S rDNA. At the lower part of this slide, you will see a bar representing 16S rDNA. It's about 1,500 base pair long. The red region represents conserved region that can be used to align 16S rDNA from different microorganisms. The blue region represents the variable region, which are usually group or species specific. They can be used to identify microorganisms down to species level. If we look at the eukaryotic microorganisms, they have 4 different rRNA, the 18S, 5.8S, 26S, and 5S. The 18S rDNA and the 5.8S rDNA and 26S rDNA are transcribed together. The 5S rDNA is made separately by a different RNA polymerase. The region between the 18S and the 5.8S is called ITS1, or internally transcribed spacer region 1. And the space between the 5.S and 26S is called ITS2. Right in front of the 18S is the ETS1, which stands for externally transcribed spacer region 1. And then behind the 26S is the ETS2. For microbial identification, scientists have used 18S rDNA sequencing. Some other scientists also use ITS1 or ITS2, even D1, D2 region in the 26S. Over the past 10 years, microbial identification by rDNA sequencing has become a fast and inexpensive alternative to traditional methods, especially the 16S rDNA sequencing. It can classify different bacteria into different species level. In addition, it was also used to describe species that have never been successfully cultured. The advantage of microbial identification by ribosomal rRNA gene sequencing over the traditional methods are they are fast and accurate. They are capable of identifying microorganisms to species level. Also, there are increasing number of sequences being added to the databases. There are multiple ribosomal RNA gene databases available. In US, we have NCBI. In Europe, we have EMBO. In Japan, we have DDBJ. And in US, we also have RDP, which stands for Ribosomal Database Project. There are also various private databases that are available to the public to use. At this point, I would like to introduce microbial identification using GenomeLab GeXP genetic analysis system. Get your results within 8 hours. This slide shows the flow through of the microbial identification using GenomeLab GeXP genetic analysis system. This workflow is very simple and fast. First, you isolate genomic DNA from your sample. Then you amplify your target in a thermocycler. After quick post-PCR cleanup, you do the sequencing on the GeXP system using the sequencing kit we provided. And then the separation and the data analysis are fully automated. The sequences are used to identify the microorganisms. Let's take a look at the workflow step by step using the 16S rRNA gene sequencing as an example. The first step is to extract the genomic DNA from your sample. There are a lot of different options you can use. For gram-negative bacteria and the Mycoplasma, a quick cell lysis is good enough. Basically, you pick up some cells from a single colony or pellet the cells from 50 microliter liquid culture that grown from a single colony, and then you resuspend the cells in 100 microliter water, place it in a boiling water bath for 10 minutes, spin briefly to precipitate the debris, and the supernatant can be used as DNA template for PCR. Alternatively, you can use magnetic beads-based extraction method, which would be suitable for both gram-positive and gram-negative bacteria. And the example we're recommending is the Beckman Coulter manufactured Agincourt GenFind V2. For fungus samples, we recommend using silica-based spin column format isolation kit. The second step is to do PCR amplification of 16S rDNA. Basically, you mix water, buffer, DNA polymerase, DNGP, primer mixture, and sample together and place them in a thermocycler. You have two options for target amplification. First, you can amplify them in full length, which will generate complete sequence for higher discriminating power in identifying microorganisms. You can also generate partial sequence that will take shorter cycling time for fast screening purpose. On the right side of this slide, you will see an aqueous gel image of the amplicons from the PCR reaction. In the center lane, we have the molecular weight marker or size standard. On the right lane, we have the full-length PCR product, which is about 1,500 base pair long. In the left side, in the left lane, you will see the partial sequence that's about 800 base pair long. The third step is the post-PCR cleanup step. Many of us know that post-PCR cleanup is a critical step for the successful implication of the downstream amplifications. The post-PCR cleanup method we recommend is the magnetic beads-based Agincourt Ampure XP manufactured by Beckman Coulter. It is very simple to use, takes less than 15 minutes to do, and it generates high-quality DNA with no primer dimers and no salt carryover. So very briefly, you add Agincourt beads solution to your sample, let it bind for 5 minutes. After DNA is bound to the magnetic beads, you place your tube or plates on the magnet for 2 minutes, and then the magnetic beads bound with the DNA will be separated from the rest of the solution, which contains the primers, the primer dimer, the DNA polymerase, and the salt. After that separation, you can aspirate the cleared solution with a pipette or a vacuum. After that, you wash the beads with 70% ethanol and leave it there for half a minute. And then you aspirate ethanol wash solution, add the elution buffer. Inside the elution buffer, the DNA will be released from the magnetic beads, and then you can transfer the eluted DNA from the tube to a new tube. So very fast, simple process. There's other options too, such as silica-based spin column format. Both Agincourt and Clonetech offers excellent spin column kits. They are excellent for isolating PCR amplicons from aqueous gel, and this is especially useful if your PCR amplicon is not pure, you have more than one band. Then you can separate them on aqueous gel and then cut out your favorite band and then extract the DNA from the aqueous gel. The fourth step is to sequence the target. Very fast, you assemble the sequencing reaction using the Terminator cycle sequencing kit from Beckman Coulter and SciEx. And then you place the reaction in a thermocycler. After a quick cleanup using clean stick provided by Beckman Coulter, then the fragments can be separated on the GeXP instrument using capillary electrophoresis. After separation and data analysis, you can take the DNA sequence to NCBI database to do a blast search to identify the microorganism. Before we move on, let's look at some important technical details. First, what are labeled dideoxy terminators? First, let's look at the normal deoxyribonucleotide structure. On the upper left corner of this slide shows the structure of a normal deoxyribonucleotide. It has three parts: the base, which can be any one of the A, T, or G, or C; the deoxyribose, which is a sugar in a ring structure; and the phosphate. For simplicity, we're showing it as a monophosphate. Usually it has three phosphates called triphosphate. Why do we call it deoxyribose? That's because deoxyribose is missing an oxygen that's present on the second carbon atom in the sugar ring in comparison to the regular ribose that's present in all RNA molecules. Notice that in the deoxyribose sugar ring, on the third carbon position, there's a hydroxy group connected to this carbon atom, and it's colored in red on this slide. This hydroxy group is very important for the formation of phosphodiester bonds in DNA. If you look at the right side of this slide, you will see the structure of DNA strand. And then you will realize that the deoxyribonucleotide is the basic unit of DNA molecules. Different deoxyribonucleotides are connected by phosphodiester bonds. DNA polymerase can use deoxyribonucleotide to grow the chain longer and longer, and based on base pairing rules with the template strand. Let's look at the structure of dideoxyribonucleotide, which is located at the lower left corner of this slide. Here you can see that the dideoxyribonucleotide structure looks almost exactly the same as the deoxyribonucleotide. However, instead of having a hydroxy group at the third carbon position, in dideoxyribonucleotide, it has a hydrogen group. Therefore, the DNA polymerase can still recognize the dideoxyribonucleotide, can still incorporate it into the growing DNA chain. However, the chain would terminate after the addition of this dideoxynucleotide because it cannot form phosphodiester bonds. Therefore, we call dideoxyribonucleotides as terminators because once they get onto the DNA, the chain terminates. The di-labeled dideoxy terminators are the dideoxyribonucleotides with a di attached to the base. If we use a different di attached to each one of the 4 different bases, by the detection of the di, we will know which base is at the end of a terminating DNA chain. Let's keep that in mind when we move to the next slide. This slide shows Sanger di-terminator cycle sequencing. It all starts with reaction mixture that's made from DNA template, the sequencing primer, the enzyme, which is a DNA polymerase, and the NTPs, which stands for deoxynucleoside triphosphate. We have 4 of them, T, C, G, and A. And we also have 4 diterminators, T, C, G, and A. Each Terminator is labeled with a unique fluorescent dye. Once we assemble this reaction, we place it in a thermocycler. So what's happening in the thermocycler? Well, we repeat a 3-step procedure 30 times. The first step is the denaturation step, in which at a high temperature, we separate the double-stranded DNA into 2 strands, the complementary strand and template strand. The second step is an annealing step. During this step, we lower the temperature so that the sequencing primer can anneal at a specific location on the template strand. The third step is the extension step, where we increase the temperature to the optimal temperature at which the DNA polymerase can perform the best. The DNA polymerase will incorporate the DNTPs into growing DNA chain and making the DNA longer and longer. However, due to the presence of Terminators, the chain could terminate at any position after the primer due to the presence of the terminator. Therefore, after 30 cycles of this 3-step procedure, we make a product mixture which contains different DNA fragments with different lengths or size. Each one of them is labeled with a dye at the end of the fragment. So for cellular sequencing, the foundation is based on the selective incorporation of Terminators or the Dideoxy Terminators by the DNA polymerase. If we separate these products, capillary electrophoresis, the analyzed data will look like the data showing on the lower right corner of this slide. Now let's look at sequencing using capillary electrophoresis. Capillaries are hollow glass tubes with a very narrow inner diameter. They are as thin as your hair. Usually the outer side is coated with a chemical called polyimide to make the capillary more durable, more strong, and more flexible. If you fill a capillary with buffer and place 2 ends in buffer vials and apply electricity, the molecules within the capillary will move based on their charge to mass ratio. In order for sequencing on capillary electrophoresis to work, we need to remember 3 important points. The first important point is that the inner wall of the capillary has to be coated to prevent the electro-osmotic flow. The electro-osmotic flow moves in the opposite direction from the direction of DNA movement. Therefore, the inner wall of the capillary needs to be coated so that DNA molecules can move in the right direction and with the right speed, and not affected by electro-osmotic flow. The second important point is that all DNA molecules they have the same charge to mass ratio. Therefore, the capillary needs to be filled with a sieving matrix to provide separation by size. So the second important point of sequencing using capillary electrophoresis is that the inside of the capillary needs to be filled with a sieving matrix. On the GeXP system, we use the linear polyacrylamide gel, which offers high resolution in separating the DNA molecules by size. The third important point is that the fluorescently labeled DNA fragments separated by size needs to be detected, so that by the presence of the fluorescent dye, we can decide which base is present at the end of each fragment. Now, with all the technical details explained, it is only logical that the GeXP system is very easy to use. On this slide, we show you 5 basic operator steps. First, you install the capillary array. Then you install the gel cartridge. In the third step, you load sample plates and a buffer plate. And then you set up your sample plate with the software and start the run. Sample separation and analysis are fully automated on the GeXP system. After the run is finished, you can come back and look at your data. Let me point out that each capillary array contains 8 capillaries, which will provide your data for 8 samples, make it fast, and then the window on the array fitting side is the side where the fluorescent detection is conducted. This slide shows how the sequencing data look like. At the bottom of the slide shows the raw data, in which on the electrophoregram you can see that smaller DNA fragment migrate faster than the longer fragment, which comes out later. On the top panel of this slide shows the analyzed data in the electrophoregram format, and that in which you can see individual dye peaks, and above each individual dye peaks is showing the bases corresponding to the peaks. And the second panel shows the core score, which stands for the base calling quality of the sequence data. And in the third box, it shows the DNA sequence for this data. Once you have the DNA sequence, you can go to NCBI website to select nucleotide blast. And then on the blast search website, you can copy and paste your assembled sequence in this top box here. Choose 16S rRNA sequence database, and then select MegaBlast, and then click Blast. You will get your results returned in a minute or two. On the upper panel shows the alignment scores, and the lower panel in the table, it shows a description of the microorganism that contains the sequence that has high homology to your input sequence. If the identity is over 99% and query coverage is over 99%, most likely this microorganism is the microorganism in your sample. Let's look at some examples. In this example, we show the sequence data from a Bacillus contaminants in our sample. In this slide, we sequenced the 18S rDNA from a yeast contaminants in a beverage sample. We also tried to type the yeast with culture method. The culture method indicated this is a wild yeast 1 or Candida cruzi, but the sequencing information revealed that this is a wild type 2, wild yeast 2 or Candida tropicalis. By doing another method, XP PCR on the GeXP system, we confirmed that this contaminant was a wild yeast 2, Candida tropicalis. This is a great example showcase the capability of the GeXP genetic analysis system in accurately identifying the microbial contaminant in the sample. On this slide, we show you the results from one of our customers' publication on International Journal of Systematic Evolutionary Microbiology. They followed a very simple step. Genomic DNA was extracted from sample, 16S rDNA was amplified by PCR. PCR amplicon was sequenced. Sequence was submitted for blast search in NCBI database, and sequence similarity information was used to generate the phylogenetic tree. And on the left side of this slide shows the phylogenetic tree that was constructed using 16S sequencing information, and a new strain, Mycoplasma amphiforme, was identified. Now let's discuss the challenges with the microbial identification using 16S rDNA sequencing. Multiple copies of 16 rDNA could be present in one bacterium, leading to multiple ribotypes for a single organism. In this case, single copy housekeeping genes such as RPOB can be sequenced to provide complementary information. Mixture of bacteria and other microbes. Sometimes your sample has a mixture that contains bacteria or fungus. In this case, terminal RFLP or XP PCR can be used for analysis of this mixture. Some microbes grow very slowly. In this case, we can also use XP PCR analysis for this sample, and we collect the sample using filtration method. Members of some genera or species have 99.5% similarities to each other for the 16S rDNA. In this case, intergenic spacer region can be sequenced to provide additional information. Ribotyping and multi-locus sequencing typing may also be useful. Traditional biochemical tests can also be used. On this slide, we show you that there are other applications that can be conducted on the GenomeLab GeXP genetic analysis system. Multiplex XP-PCR. Multiplex XP-PCR is a PCR strategy that uses universal primer technology. It can be used for fast screening of known microorganisms. MLVA is commonly used for subtyping typical foodborne pathogen by public health labs for outbreak surveillance. Terminal RFLP and AFLP are great for microbial diversity analysis and community studies. Multi-locus sequence typing and ribotyping, they are good for identification of novel microbial species. You can also perform multiplex SNP detection on the GenomeLab GeXP genetic analysis system. And the multiplex SNP detection is based on single base extension method. You can also perform multiplex STR detection. In summary, Sanger sequencing is more accurate and reliable than traditional microbial identification methods. Capillary electrophoresis analysis of 16S rDNA sequence is simple, fast, and accurate. Other molecular microbial identification and screening applications can be performed on GenomeLab GeXP genetic analysis system. That concludes my presentation, and I'd like to turn it to Judy for questions.
Excellent presentation, Dr. Luo. Thank you for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And the first question is, can the GeXP detect multiple microbial contaminants?
Absolutely, yes. With the multiple contaminants, we can use terminal RFLP and XP PCR for detection of the multiple microbial contaminants in the sample. Thank you. And next question.
We often encounter unculturable microbes such as virus. Can the GEXP be used to screen viral contaminants?
Absolutely. GeXP can be used to screen slow-growing virus. With XP-PCR, we need only a very low amount of DNA sample to perform analysis. So they're excellent for detecting the virus sample that grows slowly. Thank you. And next question, please.
If there is a mix of microorganisms in our sample, sequencing cannot work. The sample must be cultured and it may take time as well. So how can we compare sequencing approaches to the conventional methods in terms of decreasing analysis time?
That's correct. If you have a mixture of bacteria, you cannot use straight sequencing to identify them. We offer 3 options, such as terminal RFLP, the XP-PCR technology, and also you can do sequencing after you construct a DNA library with the rDNA amplicons that you made from the mixture of bacteria. When we compare the sequencing with the culture method, we usually try to compare them in terms of how fast it can identify the microorganism down to the species level. Thank you, and next question, please.
For the post-PCR cleanup using the Agincourt Ampure XP, what is the beads/DNA template ratio that is used in the process?
The ratio is 1 to 1.8. So for 10 microliter of your sample, you add 18 microliter of the Agincourt Ampure XP bead solution. If you send your email to me, I can email you the entire protocol. Thank you, and next question, please.
What are the advantages of BD Bactec over bioMérieux Vitek systems?
That's an excellent question. Actually, the Vitek method is very common and very straightforward to use. Many customers like them. Through the discussion of those customers, we know that sometimes the test results is inconclusive. The test needs to be repeated, or the microorganism needs to be identified using other method. That's why the sequencing method offers a better alternative, because you have less ambiguity when you do sequence alignment. Thank you, and next slide, and next question, please.
Okay, this next person has 2 questions for you. The first one is, have you used this technique for identification of microbial pathogens in clinical samples directly?
Yes, we have many customers who publish papers identifying the microorganisms in clinical samples. If you email me, I can send you a long list of those papers. Thank you. And next question, please.
Okay, and the next question is, in case of fungal contamination, how could you use this technique with a combination of contamination?
That's an excellent question. If you have a mixture of fungus or other microorganisms, we have three alternatives that you can choose. Either use terminal RFLP, or you can do GeXP PCR detection, or you can do sequencing using a library constructed from the amplicons during the rDNA amplification. Thank you, and next question, please.
Okay. What do you mean by GeXP PCR?
That's an excellent question. GeXP PCR is a technology that utilizes universal priming technology, in which after a few cycles of PCR, you convert multiple primer-based multiplex PCR into a PCR reaction that's based only using one pair of PCR primers. In this way, you minimize the annealing bias introduced by different primers, and you amplify all amplicons essentially using the same pair of primers. In this way, the multiplex PCR is much more quantitative and accurate over the conventional multiplex PCR. Thank you very much.
We have time for one last question. If we didn't get to yours, we will follow up with you via email. So the last question is there a limitation for identification if the microbe is isolated on a highly selective agar?
That's an excellent question. Actually, for sequencing, when we amplify the rDNA sequence, the polymerase does not really care where your genomic DNA come from. If it was growing in simple agar plate or complicated agar plates, as long as you provide the DNA polymerase with the genomic DNA, it will amplify it. So therefore, there is no limitation on identifying microbes that were growing on complicated agar plate. Thank you very much.
Thank you, Dr. Luo. Do you have any further thoughts you'd like to share with us today?
Not really. I just would like to invite people to come to our website, SCIEX website, to look at our products. And if you have any further questions, you can email me. Thank you very much.
I would like to thank our speaker, Dr. Luo, as well as our sponsor, SCIEX, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through February 2016. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.